Hi, my name is Laurie Sanders, and I'm assistant professor in the Department of Neurology at Duke University School of Medicine. My laboratory is interested in the etiology and underlying causes of Parkinson's disease. And in particular, we focus on mitochondrial dysfunction and genome integrity. Our translational work focuses on the development of peripheral biomarkers for Parkinson's disease, and that's the story that I'm going to share with you today. So the causes of Parkinson's disease are complex and multifactorial, but mitochondrial dysfunction has an established and prominent role in the pathogenesis of Parkinson's disease. Both in vitro and in vivo models of Parkinson's disease have demonstrated a decrease in mitochondrial dysfunction, and this has been a robust finding across multiple labs, including our own. In addition, human Parkinson's disease, there's been observed mitochondrial dysfunction not only in the brain, but also systemically in the blood and even muscle. So while many factors that contribute to mitochondrial function have been studied in the context of Parkinson's disease, mitochondrial genome integrity has been less well studied and has been the emphasis of our particular studies. So the way that we measure mitochondrial DNA damage is using a PCR-based method that we call MitoDNA DX. It's an assay that I developed in which you can measure when genome homeostasis is out of balance. So our cells are constantly being bombarded by endogenous and exogenous sources of DNA damage. And if your DNA repair is unsuccessful, we can detect increases in mitochondrial DNA damage. And also the opposite. If there's enhanced DNA repair or other processes and pathways, such as enhanced mitophagy, we can detect a decrease in mitochondrial DNA damage. I just have an example here of peroxide treated cells, which is a known DNA damage inducer, and this shift to the right indicates that there's an increase in mitochondrial DNA damage compared to the vehicle treated controls. And through a series of calculations, we can calculate the DNA lesion frequency. This is now adapted to a 96-volt platform, so it's medium throughput, and we can measure this in real time. And all of our data is normalized to mitochondrial DNA copy number. So what we have observed is that mitochondrial DNA damage is a shared phenotype in both genetic and idiopathic Parkinson's disease. So using rotenone models of Parkinson's disease, so rotenone is a complex 1 inhibitor of the electron transport chain of the mitochondria, and when administered to rats, mimics many of the human pathological behavioral features of human Parkinson's disease. And what we found is that in the ventral midbrain, which is, of course, is a region that degenerates in Parkinson's disease, an increase in mitochondrial DNA damage in the rotenone-treated animals compared to the vehicle-treated animals. And we observed very similar findings in vitro primary neurons. Using a different assay in which we developed so that we can detect and observe DNA damage in tissue sections, I'm showing here representative sections from a healthy control brain versus someone that had Parkinson's disease. And we labeled for tyrosine hydroxylase, which shows the dopamine neurons, TOM20 as a mitochondrial marker, the aldehyde reactive probe, which is a way that we can measure the DNA damage, and the merge on the right. And hopefully what you can appreciate is that there was a substantial increase in mitochondrial DNA damage, specifically in the dopamine neurons in those that had Parkinson's disease compared to the healthy controls. And what was sort of striking is that this increase in mitochondrial DNA damage was specific to the dopamine neurons and not in other brain regions such as the cortex. And we were able to reproduce these findings also in the rotenone Parkinson's disease model. But this increase in mitochondrial DNA damage was not specific to the brain. And in fact, when we looked at the blood in the rotenone-treated animals, we saw a very large increase in mitochondrial DNA damage that paralleled this increase in the brain. We wondered whether these findings in idiopathic models and human tissue would be similar in a genetic model. So for these studies, we concentrated on LRRK2. So mutations in LRRK2 is the most common cause of both familial and idiopathic Parkinson's disease. There are several validated pathogenic mutations in LRRK2. The most common is G2019S mutation. So this mutation, along with the others, actually share etiology and biochemistry in causing an increase in kinase activity. What's exciting about this is that there's been development of specific LRRK2 kinase inhibitors, sort of wanting to test whether this might be therapeutic. And the idea is that if you can sort of decrease this LRRK2 kinase activity back down to a normal, healthy level, that this might be therapeutic. And these are actually being tested in clinical trials currently. So we first asked the question in G2019S mutation carriers in iPSC-derived neurons, whether there was an increase in mitochondrial DNA damage. And that's exactly what we found. And when we gene edit back to the wild type sequence, that this mitochondrial DNA damage goes away. More recently, we've evaluated G2019S knock-in cells in which we observed an increase in mitochondrial DNA damage compared to the wild type controls. Similarly, in G2019S knock-in transgenic animals in the ventral midbrain region, we found a similar increase in mitochondrial DNA damage compared to the transgenic controls. And like the idiopathic data, we found a parallel increase in mitochondrial DNA damage in the of these animals. So sort of taken together, we know that the G2019S mutation causes this mitochondrial DNA damage phenotype. We've have found increase both in the brain and systemic and in the blood, and that this is a shared phenotype in LRRK2 models as well as idiopathic Parkinson's disease. So we next asked, well, is the LRRK2 kinase activity required for this mitochondrial DNA damage phenotype? And to do this, we overexpressed wild type G2019S and a kinase dead LRRK2 mutant in primary ventral midbrain neuronal cultures. And we found is that only the G2019S mutation was able to induce mitochondrial DNA damage. Using a pharmacological approach with a tool inhibitor GNE7915, we either treated prior to the development of mitochondrial DNA damage while the damage was being incurred, or even after the damage was already present. And we found that exposure with LRRK2 kinase inhibitor was able to either prevent or actually reverse this mitochondrial DNA damage in neurons. We found similar findings in an endogenous system in patient-derived cells, in that the G2019S mutation induces mitochondrial DNA damage, and exposure with this LRRK2 kinase inhibitor reversed it back to healthy baseline control levels. More recently, we've demonstrated with additional LRRK2 kinase inhibitors that even very low amounts of these inhibitors is able to reverse this mitochondrial DNA damage. In summary, we've been able to correlate mitochondrial DNA damage with LRRK2 kinase activity. And given that we can distinguish a Parkinson's disease response to these drugs from the control response, we think makes it a really good candidate as a pharmacodynamic biomarker for these particular LRRK2 kinase inhibitors. So our next question was whether LRRK2 kinase inhibitors can reverse mitochondrial DNA damage in idiopathic Parkinson's disease. Might these have greater utility and even more of the Parkinson's disease population? And so we recruited early idiopathic Parkinson's disease patients in age-matched healthy controls obtained their blood, and evaluated for mitochondrial DNA damage in a blinded manner. And we found that there was a statistically significant increase in mitochondrial DNA damage, and a very interesting distribution of mitochondrial DNA damage levels, with some with a particularly high levels of mitochondrial DNA damage. We recruited an independent cohort and found almost identical results. Again, with sort of this interesting that there's this subpopulation that's really driven by really high levels of mitochondrial DNA damage and dysfunction. We next performed an ex vivo experiment where we recruited another independent cohort of idiopathic early Parkinson's patients, obtained their blood, and then treated their cells in a tube with the LRRK2 kinase inhibitors, and asked the question, can that reverse mitochondrial DNA damage? And we found, in fact, yes. If we look at the Parkinson's disease patients in a dose-dependent manner, we saw a reversal of this mitochondrial DNA damage, though there's some that were sort of like hyper responders to the drug, while some patients didn't respond at all. And this is really different on average to the healthy control response to these inhibitors, in that some decreased damage and even some increased damage compared to these drugs. So what I'm really excited to share is that we've been part of a collaboration with multiple academic institutions across the country, in which we've asked the question, well, can mitochondrial DNA damage be detected prior to a clinical diagnosis? And so we've recruited idiopathic Parkinson's disease patients, G2019S LRRK2 carriers that have a diagnosis of Parkinson's disease, as well as those that carry the mutation but are non-manifesting carriers and do not have a Parkinson's disease diagnosis, and compared to age-matched healthy controls. And I was really excited to see that we see a similar increase and the sort of heterogeneity in mitochondrial DNA damage levels in the idiopathic Parkinson's disease cohort. Those that have the G2019S mutation with a diagnosis of Parkinson's disease also had very striking high levels of mitochondrial DNA damage. In the non-manifesting carriers, there seems to be sort of these two populations, one that sort of overlaps with controls and those that had really high levels of mitochondrial DNA damage. And it's sort of tempting to speculate that perhaps it's this subpopulation that will go on to get a Parkinson's disease diagnosis. We're very interested in following these particular patients over time. So I just sort of wanted to leave you with this idea of taking a precision medicine approach to Parkinson's disease therapeutics and clinical trials, and that evaluating mitochondrial DNA damage is just perhaps one way to patient stratify Parkinson's disease patients for particular drugs, in this case, perhaps LRRK2 kinase inhibitors, and many other drugs that may be particularly targeting mitochondrial dysfunction. So I just wanted to quickly thank my lab, the members performing this work, and my funding, and I hope that you're enjoying the conference. Thank you very much, and look forward to taking questions.